Hi everybody, welcome to CPS. I hope you all had a good week. Today, I'm very lucky to introduce Jolanta Beinarovica, who is a final year PhD student at the University of Nottingham. She completed her undergrad degree in biochemistry and biotechnology at the University of East London, where her final year project focused on the production of nematode-derived immunomodulatory proteins in bacteria. Her PhD, which she's currently writing her thesis for, focuses on designing synthetic spider silks with specific characteristics, which can be used for a whole range of applications from fabrics to antimicrobial coatings. Jolanta is also an accomplished science communicator. Her article published in The Biologist last year is definitely worth a read. Without further ado, I'll hand over.
Thank you so much. It's a very kind introduction. Let me see how to share my screen with you. Okay, just to confirm, can you see my screen okay?
Yeah, that's great.
Brilliant. Okay, hi everybody. My name is Jolanta Beinarovica, as Chiara said, a final year PhD student at the University of Nottingham. And today I will tell you a bit about the absolutely fantastic field of recombinant spider silk biotech and how we can make antimicrobial surfaces using silk and click chemistry. Now, first of all, what is spider silk and why are we so excited about it? Now, spider silks are proteinaceous fibers. They're spun by arachnids throughout their lifetime and also by various insects at their larval stage. Spiders spin up to 7 different types of silk for different applications. And major ampullate silk is the model silk in the field. So this is what I'll be mostly focusing on when I'm saying the spider silk. There's also sort of sticky structures as well as soup-producing structures and cement silks, but they are less researched and there's more work to be done in those fields if you're interested. Now, silks are of major interest in biotech due to their excellent mechanical and biological properties. Silk fiber is stronger than bulletproof Kevlar and also carbon fiber. Spider silk can be extended 3 to 4 times its original length without deformation or breaking, which means that it's very tough, which means it can absorb a lot of force before it breaks. Spider silk fibers exhibit phenomenon of supercontraction, which means that in presence of water, it gets shorter, which is the opposite what other polymers do, that get longer when they're in water. Now, lastly, spider silks have non-fibrillar nature, and that means that they can be frozen to very low temperatures before they get brittle. So as opposed to being one solid wire, it's a lot of smaller structures that gives a lot of benefits as well. Now, biologically, spider silk is very slowly biodegradable. We're talking years. And overall, it has excellent biocompatibility and it's non-immunogenic. Now, it's very rare for protein to be non-immunogenic in mammals, but this one is for various reasons. And now all these properties just come together to make silk a very appealing candidate for various biomedical applications. Now, so what's the problem here? Now, the reason why we can't buy spider silk bandages at the local pharmacy is that spiders are cannibalistic and highly territorial in nature. So if you try to farm spiders in high density like silkworms, they'll just eat each other and you won't get anything really. Now, even if you were to farm spiders, force-spun silk has inferior mechanical properties and also it's an extremely time-consuming process. For example, in 2009, 2 artists, Pierce and Godley, they created the only article of clothing made entirely of natural spider silk. And to make 4 meters of silk fabric, it took 1.2 million golden orb weaver spiders and 80 people's labor over 3 years. So obviously it's not industrially feasible. Now, as biotechnologists, we solve this problem by producing silks recombinantly in a scalable host. So we just take silk genes and insert them into something else, for example, a microorganism or a cell line or even a transgenic animal. Now, before we go into details about silk production, let's just look at silk as on a protein level. Silk fibers are composed of protein monomers, and protein monomers are called spidroins. Spidroins are very large and modular and repetitive proteins. And the sequences are quite concerned across species and also different silk types. So in essence, N-terminal domain is composed of these 5 alpha helices, and this domain allows spiders to store silk within its silk glands at ultra high concentrations, really, so up to 40% weight volume protein concentration, by forming these sort of micellar structures as nanomicelles, really. The core or repetitive domains are composed of polyalanine repeats and these GGX motifs. For X can be various charged amino acids. And these repeats is what gives silk its extraordinary mechanical properties. So the repetitive domains have not been fully crystallized. If you know anything about protein crystallization, it's almost impossible to crystallize a repetitive protein. Because this region assumes so-called liquid crystal structure. And liquid crystal is what gives silk the birefringence. So sometimes you can see silk sort of diffract light in different colors. So that's because of the repetitive domains. Now, and lastly, C-terminal domain, it provides a nucleation point for fiber formation. So it usually has unique cysteine at a very evolutionary conserved position that will form intermolecular disulfide bridge. Now, in this crystal here, you can see a dimer, so green and orange. There are 2 proteins coming together to form a dimer. And as follows genetically, spider silk genes are also huge and repetitive and have extremely high GC content, which provides a challenge for traditional molecular biology techniques to do, to manipulate further. But those luckily have been mostly resolved by either very clever bioengineering or synthetic biology. So this little cartoon just really shows this normal workflow that we have. We study interesting spiders, and interesting is whatever it makes, whatever it can be, really. So you can have diving bell spiders, and their silk is interesting because it's gas permeable but watertight. So diving bell spider makes this sort of balloon around itself and goes diving underwater. There is, for example, Darwin bark spider, known to have the strongest silk to the date discovered. But further, we sort of study silk genes by excising spider silk glands from the spider and deep sequence the RNA within those glands. And then we do a lot of bioinformatics and we design miniature silks. Now, the reason for making miniature silks is that microorganisms can't really cope with these 250 kilodalton proteins. So we need to make them smaller. And then we just express them in a scalable host and purify, and there you go, spider silk protein. In literature, silk has been produced in various hosts, from E. coli to salmonella, yeast, insect and mammal cell lines, various plants, as well as mice. You might have heard of goats. So goats express spider silk in their milk. So it's Randy Lewis's group, I believe. And more recently, there was a report about sheep embryos that express spider silk under a keratin promoter. So they would have wool that's made of spider silk. But we'll see how it goes. Okay, so in nature, silk exists either as a fiber or as a glue. There is nothing much in between. In contrast, recombinant spider silks can be processed, or sometimes they self-assemble into a range of very useful morphologies that you can see here. So we can make fibers by either electrospinning, which is just applying electrical current to concentrated protein solution and just spins it into a fiber. We can do things like extrusion coagulation, where we just extrude silk protein into a solvent bath and it just makes a fiber. Or we can design silks that assemble into fibers themselves. And so the one that you can see in the picture here is actually self-assembled fiber. Now, spider silk in solution self-assembles into very stable and continuously accumulating films that are very good at coating pretty much everything, and particularly hydrophobic surfaces, for example, plastic. And they're useful for things like two-dimensional cell support. Now, if you imagine artificial skin or anything other that's like flat. Silks also have this property of gelation, so they make hydrogels that are water-swollen polymer networks. Hydrogels are very popular 3D cell support. For example, if you have a silk hydrogel that's seeded with liver cells, and you basically have a 3D structure of a liver, potentially, maybe. Not yet, really, but you could look into that. We can also make aerogels, and aerogels are very similar to hydrogels, but except in that they have air in the pores instead of water. And lastly, you can also make a range of colloidal drug delivery systems, for example, spheres. And our spheres are very good to sort of deliver drugs in living systems. For example, if you have a spider silk sphere that has an antibody tag that will go to a tumor or to organ of choice and deliver the drug where it's needed as opposed to just everywhere. Okay, now, apart from very exceptional mechanical properties, silk really doesn't do much else. It's a bioinert protein. Now, to imbue silks with properties that they would not ordinarily have, they can be functionalized. And there are two main approaches for silk functionalization. Now, you can do it genetically, or you can do it chemically. Now, genetic fusions are called chimeras, and to make a chimera, you just genetically fuse protein with another bioactive protein in so-called upstream fashion. And if you would like to functionalize protein chemically, you would be adding functional domains post-translationally or in a downstream fashion. Now let's look into more details and sort of little pros and cons of each. Now, just to highlight, at this point, we are looking at silk as a scaffolding for delivering a desired functionality. So it provides you the structure, but other than that, it doesn't do much else. Okay, so in genetic functionalization, we create brand new proteins, these brand new chimeras. And there's been a wide range of silk chimeras reported in literature, and they deliver such wide range of functionalities. For example, you can fuse silk with things like cell penetration domains or gene delivery domains, cell adhesion motifs, tumor recognizing motifs. There are proteins that naturally chelate metals, for example, silver, and also various antimicrobial peptides and antibiofilm peptides, for example, silk and human neutrophil defensins, and it will deliver the functionality. Now, this approach is very appealing because of its reproducibility. The expressed protein will always be the same, and there will always be one functional domain or the designed amount of domains per one silk molecule. But there are a number of limitations. Now, firstly, in this fashion, we can only use functional proteins, so only proteins and not any other type of molecules, really. From my sort of bio manufacturing perspective is that each chimera is a brand new protein, and it will require cloning. It will require optimization of growth and processing conditions. So it's time consuming to develop brand new chimera every time. Also, depending on where the genetic fusion is located, it can have deleterious effects to silk's mechanical properties. For example, C-terminal fusions often fail to make spontaneous fibers, and fusions in the repetitive domain impair the strength of the fiber. Like I mentioned before, the repetitive domain is what gives silk the strength. And finally, it is just challenging to make antimicrobial materials in microbes because obviously the protein will kill the host, and it results in low yields and poor quality of the material. So, to avoid the aforementioned limitations, we can modify silk after it's been translated using bioconjugation. Now, I'm pretty sure that all chemists in attendance recognize this reaction here. It's called copper-1 catalyzed azide-alkyne cycle addition. But for those of us who aren't, I'll go into a bit more detail. So what we do is we feed bacteria a synthetic amino acid analog that has a selectively reactive side chain. For example, instead of methionine, we feed bacteria with azidohomalanin. And it has this azide side chain, so these N3 side chains. And now we can use these chemical groups to conjugate molecules of interest. Now, azides will react with terminal alkynes in very mild one-pot conditions in presence of copper to form a stable triazole bond. And we have exploited this reaction to conjugate things permanently, so they are not designed to be released from the surface, but also in a way that allows molecule to be released from the surface. Now over here in red, I've highlighted the ester bond. And ester bond is obviously can be hydrolyzed using esterases or just low pH. So that bond breaks and releases an active molecule into the environment. Now this is necessary because, for example, for this particular molecule, levofloxacin, levofloxacin has an intracellular target. So it needs to go away from the surface into the environment and be taken up by bacteria where it exhibits its antimicrobial effects. And these are a couple of data sets from previously published work. In the reaction shown in the previous slide, levofloxacin has been conjugated to silk fibers. And those silk fibers show sustained and significant antimicrobial effect against E. coli, and this antimicrobial effect was sustained over 7 days, which strongly suggests that the release of antibiotic happens in response to bacterial growth as opposed to just being, just randomly diffusing away. Now, in our group, we also have conjugated spider silk films and fibers to RGD cell adhesion peptide. And those peptides were designed not to be released, they were permanently attached to the silk. And these conjugates significantly promoted cell adhesion compared to non-functionalized fibers and even sort of standard tissue culture plastic. Now, I can't tell you too much about my own work because it's all unpublished, but what sort of my PhD is focused on is to expand the library of conjugates. We want to see how many molecules we can attach, what are their functionalities, and sort of move away from small molecules to sort of organic polymers and also other biopolymers to expand sort of the portfolio of functionalization potential, really. Now, I always like to briefly touch on this. Now, silk biodesign fits into this sort of broader area of biodesign, where people use biological systems and synthetic biology to produce various products for consumers. So you can see here that the guys, so the tie, the dress, and the parka, they're all made of spider silk. And there is a moisturizer, there is a cell scaffolding made of spider silk. So this is this red foamy sort of structure. And the white blob in the corner is a breast implant. Breast implant was coated with spider silk to reduce silicon's immunogenicity, because sometimes some people develop reaction to silicon, but they will not develop reaction to spider silk because from previous evidence we know it's just not immunogenic, which is great. And there's many ways we sort of employ biology these days to use to try and provide sort of consumer benefit, and these designer proteins help us to do so. Very brief acknowledgements. So my boss is Professor Neil Thomas, and my second supervisor is Sarah Goodacre. The group lead is David Harvey, and under his surname you can find the publications that I was speaking about earlier. We have a team of organic chemists that sort of modify the ligands and allow us to test them, and University of Nottingham imaging facilities, and also BBSRC UKRI, who fund my research. That's all from me for today. I still have a lot to tell you, but obviously we are limited on time. But you can read a little bit more about sort of industrial relevance of silk proteins and biodesign in my article in The Biologist. It's called Futuristic Fibers. I think it was published in June, I believe, this year. And if you have any questions or sort of any ideas, I'm happy to take them now. And please do contact me via your preferred channel. As this slide says, I'm unemployed from September this year. So if you have any projects that you need a synthetic biologist on, I'm available. Thank you very much. And over to you.
Thank you so much. That was such an interesting talk. By the way, I have so many ideas. Let's open the floor for some questions. If you'd like to put your hand up and then unmute, or if you'd like to type them in the chat, I can read them out. Timothy, do you want to unmute?
Can you hear me now?
Yes.
Yeah, I can hear you.
Right, yes, thanks for a very interesting talk. I was wondering how much of your antibacterial substance you can, conjugate to the silk protein that's produced. And one of the challenges, as I'm sure you know, in antibacterial materials is keeping an antibacterial effect going for a longer period of time. So could you comment on that if it's not a secret?
It's not a secret. It's actually been published in this paper, in this levofloxacin paper that I was talking about. It's called Antibiotic Spider Silk. So if you Google that, it will come up. Now, the controlled release of antibiotic is obviously a key theme. And this is mostly achieved by very clever linker design. So in chemistry, some bonds hydrolyze faster, or some bonds are responsive to a signal. You can break a bond with light, with enzyme, with acidic environment, and it makes these sort of clever, sort of smart release systems. So the ester bond will only break when the pH of the environment drops. And decrease in environmental pH is something associated with bacterial growth. Now imagine bacteria growth, they produce growth and they produce acid and, well, lactic acid really, and the bond breaks and releases the antibiotic. Now that's the to answer the second part of your question. To answer the first part of your question, so how many molecules of antibiotic we can conjugate to a silk protein? The protein that I work on, it has 3 methionine residues in it. I've designed it so. So it can carry 3 antibiotics per molecule of silk. But as you can see, it can be modified. So if I would like to have 6 methionines, I can just engineer them in, and my silk will be able to take 6 azidohomalanines and therefore provide 6 azido handles for conjugations and carry 6 molecules of levofloxacin, for example. I hope that makes sense.
Thank you very much. We have a question in the chat that somebody asks, are there any disadvantages to using spider silk? And if there are, what can be done to improve on them?
So one of the disadvantages that we face as researchers is obviously scale. If you were to make a kilo of spider silk, you need a massive scale of plant. And at this point, unnatural amino acid incorporation is something that's not assessed on a kilogram scale. So that's one fairly big disadvantage. Yields of protein, when you try to incorporate unnatural amino acid, tend to be significantly lower. For example, in a shake flask culture, without incorporation of a synthetic analog, I can get hundreds of milligrams, whereas if I add a synthetic analog, I get tens of milligrams. So that's the reduction that you can see. This is because bacteria will take up the synthetic analog, incorporate it into native proteins, and eventually it will have detrimental effects, thus lower yields. But there is something called amber codon and amber codon engineering, and that sort of tackles this issue with my particular approach. And that's, again, very clever genetic engineering, but this is something to be sort of investigated further. Yeah, that's probably one main disadvantage, I would say.
Thanks very much. Ruben has a hand up.
Hello. So something you mentioned in your talk is obviously like spider silks are extremely high molecular weight proteins, and obviously trying to produce these in like say E. coli, obviously you need to reduce the molecular weight. Have you found that reduction in molecular weight has any kind of negative impact on the spider silk properties, or?
Yeah, that's a very good question. I probably didn't mention that. So what we know from the research and from designing these sort of miniature spider silks is that the mechanical properties of spider silk are directly proportional to the length of the repetitive domain. So if you have 4 repetitive domains as opposed to 200 repetitive domains, it will obviously be 250 times weaker. So we find this direct proportionality. And on the other hand as well, there's been evidence in literature that if you can make silks double the size of what's normally seen in nature, they'll be twice as strong. So we see this direct sort of proportionality between the repeats, these alanine and glycine rich repeats, and the mechanical properties of the fiber. Next.
Thank you very much.
Neil Hawley, would you like to go next?
Hi there, how you doing? I thought that was a fantastic talk, actually. Really highlights what a lot of hard work that you've done. So you've highlighted that you're working in bacteria, and obviously I think there's a lot of literature about the goat's manufacturing of spider silk. Why did you choose bacteria as opposed to one of the other systems, or what's better about working in E. coli?
So E. coli has the shortest generation time.
Yeah.
That's one of the main advantages. Second advantage is that E. coli has been engineered to be methionine auxotropic. So it means it doesn't produce its own methionine. And that forces the bacteria to take up the synthetic analog that it may or may not quite like, but it looks just about the same. So it will force bacteria to take it up. Now, one step further, I also like working with yeast, but spider silk have some sort of secondary protease site in the C-terminal domain, and we find that if you produce silk in yeast, the quality of the silk is sometimes lower, and it may or may not spontaneously form fibers. So if you want to make fibers, it's not ideal. Now, spider silks have been produced in mammalian cells and insect cells, but because of oxide environment within these sort of animal cells and eukaryotic cells, is that silks will spontaneously form fibers within the cell, eventually killing it. Now, once a silk has formed a fiber, there's nothing much you can do. It's almost impossible to dissolve a fiber, which is very, it's very exciting, but also not that great if you want to work with soluble protein. Again, when it comes to the goats, I read a bit of literature about these goats and how they were made and how they purify the silk. But you need to bear in mind that milk is very protein and fat-rich environment. So purification of spider silk from milk is not as straightforward as they thought it's going to be. So there's always difficulties. But yeah, as I said, E. coli heavily engineered to be sort of a good host with short generation time. So I'm going with that for now.
Okay, good. Could I ask one more question, if that's okay?
Yeah, of course.
I think
Go
you
for
might have
it.
alluded to it already. So have you looked at codon bias, altering this sequence so that you biased it better towards manufacturing in E. coli rather than, because obviously you're taking a spider silk and trying to produce it in E. coli.
Yeah, it's a very good question. Yeah, so we have looked into that, but what we've also found is that silk's transcription and translation, if there is a rare or sort of less preferred codon in it, it's there for a reason. Because if you have these repeats, these 16 to 18 alanine repeats, and polymerase and the ribosome sort of tries to read all of them, because of how ribosomes are, they don't like repetitive proteins, so they kind of come off. So if we see a rare codon, it's very likely it's there for a reason. Now, in spiders, what spiders have done, they have evolved redundant copies of certain tRNAs that will bring amino acids to the silk where there is sort of certain requirements. So they have larger tRNA alanine and tRNA glycine pools to sort of to compensate for these requirements. So I think there is definitely some engineering to do, but with very sort of top level approach that we've seen is that, yeah, if there is a rare codon, it's probably there for a reason.
Okay, that's fantastic. I think you'll have no problem getting a job when you finish your PhD.
Thank you, yeah.
Definitely agree. I have a final question about when you make the proteins from E. coli, if you're making antimicrobial ones, do you have a problem that they do kill the host, and do you have to have a sophisticated way of extracting the protein as it's synthesized?
See, this is the beauty of modifying silk post-expression. So bacteria will just make plain protein that doesn't really engage with living system. It just kind of just floats around and, you know, sometimes forms a fiber or two, and that's it. Now, then what we do then, we pop them open and purify the silk. And from there, we treat silk protein as a chemical. So there is no more living systems involved. So that's sort of more streamlined approach, which is yet another sort of benefit of chemical approach of functionalization. If you were to make a chimera of silk and an antimicrobial peptide, yields ultimately are very, very low. You'll get single milligrams of protein per shake flask, for example.
Thank you very much. That makes sense. We've had a few more questions in the chat, if that's okay. So somebody asked if it's possible to use spider silks in defense and space applications in the future.
That's a good question, and I think that one of the companies that's called a Japanese company, Spiber Inc. I think there have been some reports of them using the military funding for their purposes. In the literature, I have not seen any evidence for that, but obviously it's going to be sort of the industrial side of it. Now, interestingly, what we found is that, well, not me, but researchers from Oxford have found that silks survive well in very cold environment because of this nanofibrillar nature that I touched upon. Now, so that means that there could be applications in sort of space and military.
Thank you very much. One last question is, does the replacement of alanine in the repeating unit section with azidohomoalanine, does it affect the mechanical properties of the silk produced?
Right, so azidohomoalanine is an analog for methionine. So we do not replace alanines with anything because that's guaranteed to have detrimental effects. Now, methionine is a relatively rare amino acid in all proteins. We usually think it's very abundant because it's the first one, but actually it's relatively rare. And yeah, silks, the C-terminal domain on the construct, that's one of my favorites, that's derived from Uprocenops australis, which is the nursery rare spider. It has only 3 methionines in the C-terminal domain, which doesn't sound a lot when you take into consideration a 250 kilodalton protein. And we use that to replace these 3 methionines with azidohomoalanine and then use them for conjugation. And we have not seen a detrimental impact on silk's mechanical properties compared to sort of wild type, where all methionines would be actually methionines as opposed to azidohomoalanines.
Thank you very much. That's all the questions. I just want to say a massive thank you from the Society for an absolutely incredible talk. It was a perfect summary of your research, and wish we could show a round of applause.